Hello, my name is Joshua Gabi. I welcome you to Coordination and Control. Questions 21 to 25. State the comparison between nervous and hormonal control in animals. Part B. Explain the mechanism controlling the release of hormones by glands. Part C. Outline roles of coordination and control system in living organisms. State the comparison between the nervous and hormonal control in animals. We shall start with the similarities. Both release chemical substances as a means of communication between cells. In both, their principal role is coordination and control of the major physiological activities of the body, thus maintain a steady state in an organism. Now let's look at their differences. Nervous control, the electrical impulses and chemical transmission. Whereas hormonal control, it is only chemical transmissions, which are hormones. Nervous control, it is rapid transmission and response. Hormonal control, slower transmission and relatively slow acting. Adrenaline is an exception. Nervous control. This offers a short-term changes or effects. Hormonal control offers long-term changes. Nervous control response often very localized, and in hormonal control, the response may be very widely spread. Nervous control, the pathway is specific through nerve cells. Hormonal control, the pathway is not specific. That is, hormones circulate in the blood stream to specific target sites. Nervous control. Origin of the stimulus is sense receptor or organ. Hormonal control. The origin of the stimulus is gland. Nervous control. Destination of the stimulus is to specific point. Hormonal control. Destination is all over the body. Nervous control. Site of action is the effector, that is the muscle or gland. Hormonal control. Site of action is the target organ. Part B. Explain the mechanism controlling the release of hormones by glands. Presence of specific metabolites in the blood. For example, excess glucose in blood causes the release of insulin from the pancreas, lowering blood glucose level. Presence of another hormone in the bloodstream. The release of trophic hormones depend on the presence of the releasing hormone or inhibiting factors secreted by the hypothalamus. Stimulation by the autonomic nervous system. For example, adrenaline and noradrenaline are released from the adrenal medulla in response to nervous stimulation arising from situations of fear, anxiety, stress, and danger. Influence of the internal biological control. Release of the hormone like gonadotropins are under biorhythmic control of the internal biological clock. Outline roles of coordination and control system in living organisms. Effective cellular communication in multicellular organism. 2. Effective reception of stimuli by the receptors. 3. Effective transmission of sensory impulses to the central nervous system. Comprehensive and effective interpretation of stimulus by the central nervous system. Tailor an appropriate response to a given stimulus through the peripheral nervous system. Control body's physiological and metabolic processes, maintaining the body's steady state. Permit rapid escape of the organism from life-threatening stimuli. Number 22. Distinguish between spatial summation and temporal summation. Number 2. Apposition eye and superpositional eye. Number 3. The mammalian eye and an arthropod compound I. Compare the two. C. Explain the functions of the auxins in plants. Distinguish between spatial summation and temporal summation. Spatial summation is when multiple postsynaptic potentials, excitatory or inhibitory, from different presynaptic neurons add up to reach threshold and generate action potential that fires an impulse in the postsynaptic neuron. While temporal summation is when a high frequency of action potentials in a single presynaptic neuron elicits the postsynaptic potentials that overlap and add up with each other to reach threshold, causing firing of an impulse in the postsynaptic neuron. Distinguish between apposition I and superposition I. Apposition I is a compound I in which each ommatidium focuses only rays that are almost parallel to its long axis, so that each forms an image of only a very small part of the visual field. The image of the whole results from the combination of these part images, e.g. in Juno insects. Whereas superposition I is a compound I in which the sensory cells of an ommatidia can pick up light from a large part of vision field, so that the image received may overlap those received by as many as 30 neighboring ommatidia. The superposition image has brightness but low in sharpness compared with a position image, e.g. in nocturnal insects. Compare the mammalian eye and arthropod compound eye. We shall start with the similarities. Both have lenses and both have light-sensitive cells. Let's look at the differences. The mammalian eye, the lens is elastic, whereas the arthropod eye, the lens is inelastic. Mammalian eye has a varied focus. Arthropod eye has a fixed focus. The mammalian eye has a movable eye due to muscle attachment. Arthropod eye is immovable because of no muscular attachment. The mammalian eye has a high resolving power in bright light. The arthropod eye has a low resolving power. The mammalian eye has one visual unit. The arthropod eye has several visual units, eyelets called ommatidia. The mammalian eye has both the cones and the rods. Arthropod eye has only the cones. Mammalian eye is capable of color perception. Arthropod eye is incapable of color perception. The mammalian eye has low visual acuity due to fewer cones. Arthropod eye has a higher visual acuity due to numerous ommatidia. Mammalian eye has a lower sensitivity to light due to single eyelid. Arthropod eye has a high sensitivity due to numerous eyelids. The mammalian eye forms a single non-overlapping image. Arthropod eye images overlapping with poor quality of vision. Mammalian eye has a narrow field of view. The arthropod eye has a wider field of view. Explain the functions of auxins in plants. Part C. Cell elongation. Auxins cause elongation of cells just below the tip of the shoot or root, resulting in growth. Fruit growth. The auxins promote the enlargement of many fruits by causing cell walls of the fruits to stretch in all directions. induce parthenocarpy. The auxins do promote the development of roots without fertilization. Root initiation. Auxins are regarded as rooting hormones because they promote root development when applied to stem cuttings. Apical dormancy. This allows growth of the apical terminal buds and inhibits growth of the lateral buds. Suppression of abscission. Antagonize the effects of abscisic acid, thereby preventing premature leaf fall or fruit fall. (23) Explain why the eye of the cat glows at night when shone with a flash of light. Part B. Explain the trichromacy theory of color perception. Part C. How does the human eye adjust itself in order to focus a near object? Part A. Explain why the eyes of the cat glow at night when shone with a flash of light. Lying immediately behind the retina of the cat is a shiny layer called tapetum lucidum that refracts visible light back through the retina, increasing light available to the photoreceptors. Tapetum lucidum contributes to the superior night vision of cats. Tapetum lucidum functions as a retroreflector, which refracts light directly back along the light path, maintaining the sharpness and contrast of the image on the retina. The refraction also occurs by constructive interference, thus increasing the quantity of light passing through the retina. Hence, the sensitivity of the cat's eye to light increases. Explain the trichromacy theory of color perception. According to the trichromacy theory, color perception iodopsin, the photosensitive pigment in the outer segment of the cones, exists in 3 isoforms, each occurring in one of the three cones and is sensitive to either red, blue, or green light. Differential stimulation of red, green, and blue cone by light of a particular wavelength leads to the bleaching of the corresponding isoform of iodopsin, establishing an action potential. Information is relayed to the brain, which interprets color corresponding to the wavelength of the stimulant light. For example, 100% green, red, or blue cone stimulation leads to the perception of green, red, or blue color, respectively. Secondary colors are perceived when there is equal stimulation of any two of the cones, while equal stimulation of all the three cones yields white. Black is perceived when there is stimulation of none of the cones. Part C. How does the human eye adjust itself in order to focus a near object? The ciliary circular muscles will contract. The radial muscles will relax. The tension in the suspensory ligament is released. The lens becomes more convex, thicker, and more curved. Refracting diverging light rays form a point object, forming a sharp image of the near object on the retina. State 4 differences between electrical synapse and chemical synapse. Part B. Describe how an impulse is transmitted across a chemical synapse. And part C. How acetylcholine, one that excites skeletal muscular activity, will inhibit heart rate. Part A. State 4 differences between electrical synapse and chemical synapse. Electrical synapse, they have shorter synaptic cleft. Chemical synapse have longer synaptic cleft. Electrical synapse, the transmission is mediated by an ion current. that flows over the synaptic cleft. In chemical synapse, the transmission is mediated by chemical neurotransmitters over the synaptic cleft. Electrical synapse transmission is faster. In chemical synapse, there is slow transmission. Electrical synapse, bidirection transmission occurs. Chemical transmission, synapse, it is a unidirection transmission. Electrical synapse, longer refractory period, thus delay in the transmission. Chemical synapse, there is shorter refractory periods, thus no delay in the transmission. Part B. Describe how an impulse is transmitted across a chemical synapse. The action potential arrives at the axon terminal, causes depolarization of the presynaptic membrane. The calcium voltage gates channels open following the calcium ion influx. Calcium sensing proteins on the vesicles bind with the calcium ions, activating the vesicles which migrate to the presynaptic membrane, fuse with it, and release the neurotransmitter by exocytosis. Calcium ions quickly diffuse back to the synaptic cleft. Neurotransmitter diffuse through the synaptic cleft and then binds to specific receptors on the postsynaptic membrane, which are ligands-gated ions channel. These open, causing differential ionic exchange, resulting in the graded potential. Excitatory neurotransmitters like acetylcholine makes the postsynaptic membrane for an excitatory synapse more permeable to sodium ions than potassium ions, depolarizing it, hence forming an excitatory postsynaptic potential. So let us try to look here and we see what we are talking about. So this side, this is the whole of our synapse. This is a synapse. And we want to see how the impulse is going to be transmitted across a chemical synapse. Now, this side is the presynaptic neuron. And we have a gap here, which we call the synaptic cleft, what we are calling the synaptic space. And this side, we have a dendrite. And this side is what we call the postsynaptic neuron. So here, this membrane here is what we call the presynaptic membrane. And this one, this membrane, is what we call the postsynaptic membrane. Now, we are saying when the action potential comes here, it is going to depolarize the presynaptic membrane, and it will cause the opening of the calcium gated channels, which are going to allow the calcium ions to enter from the synaptic space into the presynaptic neuron. When these calcium ions enter, they will be attached to the receptor, calcium receptors on the vesicles. And when they attach themselves on the vesicles, they are going to cause the vesicles to move towards the presynaptic membrane. And when they come to the presynaptic membrane, come into contact with the synaptic membrane, they are going to release the transmitter substance like acetylcholine by exocytosis into the synaptic space, or what we are calling the synaptic cleft. Then the calcium now will flow out very fast, goes back to the synaptic space. The neurotransmitter now, which has been thrown into the synaptic space, will diffuse toward the postsynaptic membrane. And when they reach the postsynaptic membrane, they are going to touch onto the receptors on the postsynaptic membrane. And they are going to make this membrane to become more permeable to sodium ions. And when they become more permeable to sodium ions, they will allow sodium ions to enter into the postsynaptic neuron, and this is going to depolarize the postsynaptic membrane. And we are going to form what we call the excitatory postsynaptic potential on the postsynaptic membrane. Inhibitory neurotransmitters like GABA, glycine, cause opening of the crawling channels of the postsynaptic membrane. Hypopolarization occurs, forming an inhibitory postsynaptic potential. IPSP. Reuptake by presynaptic neurons. Enzymatic degradation and diffusion reduces the neurotransmitter levels in the cleft, determining signals. Now here, sometimes the transmitter substance that we have can be inhibitory transmitter substance. Before, we had the excitatory neurotransmitter substance, which was acetylcholine. But here now, if the vesicles contain the inhibitory neurotransmitters like GABA, glycine, now when these ones are released into the synaptic space or synaptic craft, they will diffuse and go to the postsynaptic membrane, and they are going to cause the opening of the choroid channel of the postsynaptic membrane. This will allow the entry of the choroid ions into the postsynaptic neuron. And when the choroid ion enter into the postsynaptic neuron, they are going to hypolarize the membrane, meaning that it becomes more negative. And when hyperpolarization occurs, we form now what we call an inhibitory postsynaptic potential, meaning that it cannot allow any passage of the impulse across the postsynaptic neuron. Part C. Explain how acetylcholine, one that excites skeletal muscles activity, will inhibit the heart rate. The acetylcholine released at the vagus nerve ending greatly increases the permeability of the fiber membranes to potassium ions, which allows the rapid leakage of potassium out of the conductive fiber. This causes an increased negativity inside the fiber, an effect called hyperpolarization, which makes this excitable tissue much less excitable. This decreases the rate of rhythm of the sinus node, as well as decreasing the excitability of the atrioventricular junctional fibers between the atriomusculature and the atrioventricular node. This therefore slows the transmission of the cardiac impulse into the ventricle. The same acetylcholine has excitatory effect on the skeletal muscles. When the acetylcholine binds onto the specific receptor molecules on the postsynaptic membrane, it causes the sodium gates to open, while the choroid gates and potassium gates to close. Sodium ions diffuse rapidly inside the sarcoplasm, and inside the membrane becomes more positive, while the outside more negative. The membrane is depolarized, resulting into the excitatory postsynaptic potential, which increases to reach a threshold value, and action potential is generated. With examples, distinguish between synergism and antagonism. Part B. Outline the commercial uses of plant growth substances. Part C. Describe the role of estrogen in the menstrual cycle. Part A. With examples, distinguish between synergism and antagonism. Synergism is the interaction of two or more growth substances, those are hormones, to produce an effect that is greater than the individual effect of each hormone. For example, auxins and gibberellin interact synergistically to bring about internode elongation. While antagonism is a condition in which the interaction of two or more growth substances or hormones produce opposing effect to each other. For example, gibberellin breaks seed dormancy and thus allows germination. But the presence of abscisic acid possesses a stimulatory effect on gibberellins and thus promotes seed dormancy. Part B. Outline the commercial uses of plant growth substances. Synthetic auxins. These are utilized as rooting agents on the stem cards for the development of the adventitious roots, and control the amount of auxin utilized as selective weed killers in cereal crops. Auxins are utilized as anti-abscission agents, preventing premature leaf and fruit fall. Auxins have been used to delay flowering until an appropriate time. Auxins have been used to increase the fruiting size. Auxins are utilized as anti-sprouting agents in potatoes, therefore prolonging their storage time. Gibberellins and auxins are utilized as fruiting agents like parthenocarpic. Gibberellin is utilized as a germination promoter. Gibberellin is utilized as a bolting agent in rosette plants like cabbages. Gibberellin is utilized as an internode elongating agent in genetically dwarf plants. Gibberellin is utilized as a modifier of sex of flowers as it promotes male flowers than female flowers. Ethylene is utilized as a fruit ripening agent. Abscisic acid is utilized as a germination inhibitor or dormancy promoter. Abscisic acid is utilized as a sex modifier of plant flowers as it promotes female flowers. Cytokinins used on vegetables to preserve the green color, anti-aging agent in plants. Hence, important in prolonging the life of fresh leaf crop such as cabbages, lettuces, etc. Part C. Describe the role of estrogen in the menstrual cycle. It ensures reepithelialization of the endometrium, vascularization, repair, and preparation of the uterus for implantation. Small amount of estrogen inhibit the production of follicle-stimulating hormone and luteinizing hormone by negative feedback. Thank you very much for participating in coordination and control. Questions 21 to 25. Please do like, share, and subscribe.